Okay, good morning students. Hope you all revised our yesterday's anatomy session. Today we are going to step into a world where light meets life. Yes, the cellular story of vision. Our learning objective of the session is to decode the retinal cellular symphony and identify how each of them plays a vital role in turning light into sight. So, we are going to have this session in a triple take. First we will have a visual hook, then we will meet our retinal squad, that is our cells, then we will have a small take-home nuggets. We know that when light hits into the retina, it is the rods and cones. They are the first detectives of the light, will sense the light and they will convert this light energy into electrical energy by means of phototransduction and carry this information to the brain via the optic nerve where this is being interpreted. I know that you all are thorough with the 10 layers of the retina and how these cells are arranged in the retina from our yesterday's class, isn't it? So with that knowledge, we are going to meet our squad. Yes, here they are, our retinal squad, which each of them having peculiar function. You just imagine our eye as a visual camera, which is taking a picture and sending it to the brain. And we know that while taking photograph in the mobile phone and camera, we need a good coloring system, good contrast system, isn't it? So in our story, our cells are playing all these roles. So rods are the night watchers, cones are the color artists, horizontals are the contrast controllers, bipolars are the messengers, amacrines are the motion detectors, Muller and RP are the support squad, and ganglions are the reporters. So first we can discuss about our detectives. They are the rods and cones. Rods are responsible for our scotopic vision. Yeah, they are very much sensitive to dim light and they will provide our peripheral as well as night vision. Coming to the cones, they are responsible for our photopic vision, that is vision during bright light. They will also provide color vision as well as color discrimination. And remember, and they are very much concentrated in our fovea centralis. Coming to the bipolars, they are the messengers. You can see the bipolars is called as the first order neurons of our visual pathway. You just think of them as the post offices of the retina. Why? Because they will collect, refine and process the visual messages from the photoreceptors and transmit it to the ganglion cell. And they will make sure that the visual message is meaningful before it reaches further. Coming to the ganglions, they are the reporters. They will collect the refined information from the bipolars and their axons will form the optic nerve and carry this information to the brain. And some of them are even photosensitive themselves. So they are also involved in pupillary right reflex and regulating the circadian rhythm. And they are the final checkpoints before our vision becomes a perception as a second order neuron. Coming to the horizontals, they are the contrast controllers. Before that, can anybody tell me which phenomenon is shown in this picture? Anybody? Yeah, good. Lateral inhibition. Lateral inhibition is a clever process by which the horizontals will sharpen the edges of the images, enhances the contrast and makes the image stands out by inhibiting the impulse transmission across the bipolars on either side of the photoreceptor which is being stimulated by the light. Okay, then coming to the amacrine. They are the motion detectors. These are the ones which are responsible for bringing timing as well as movement into our story. And they will integrate the signals between the bipolar and the ganglion cells, especially when the visual scene changes very quickly, like we are seeing a moving car, a blinking of a light. Okay, coming to our last, Muller cell. They are the support squad. You can see the Muller cell is giving various supporting hands to the retinal cells, isn't it? So it is a supporting glial cell. It is involved in metabolic support by means of ionic balance and regulating the neurotransmitter, removing the waste products, etc. And they can be called as the doctors of the retina. Why? Because they are involved in retinal repair and regeneration after an injury like a doctor. So coming to our last layer, that is our guardian security layer behind the photoreceptors, that is RP. It forms a barrier between the retina and the choroidal blood vessels, that is our outer blood retinal barrier. Okay, they are also involved in phagocytosis of the photoreceptor disc and recycling of the visual pigment. And what are the take-home nuggets? What points you should remember? Yeah, these are the key words you should take from our class, okay? And remember one thing, retina is not at all a passive screen, but an active processor containing living circuit of cells. Together, they transform mere photons of light into the rich experience of our vision, okay? Now we have met the team behind the vision. Tomorrow, we are going to trace the path of sight together, that is visual pathway. See you tomorrow. Thank you.
Still have to, they have doubts, madam.
Yeah, we can clear.
Madam, I have a doubt.
Yeah.
I have seen some children with dystrophy, they said, ma'am. They keep poking their eyes. I don't understand why they're doing that.
Poking.
Huh.
Maybe they have some dystrophies related to rods
But
and
why are
cones.
they poking, ma'am?
Okay, I'll refer and count. Okay. Maybe they have some degenerations or dystrophies related to rods and cones.
Like they said LCA is the diagnosis, but
I-
the child kept poking the eye. They said Leber's congenital amaurosis is the diagnosis, but the child kept poking the eye and the eye was also looking sunken. I don't know why.
Okay, that is a good question. I'll refer back you and tell you, okay?
Thank you.
Ma'am, you just taught us that macrine cells are support staff. Do they have any other function?
Which cells?
The amacrine cells.
Amacrine cells.
Yeah, the other.
Yeah, they perform a peculiar process called temporal modulation.
Okay.
By means of they modulate the responses between the bipolar and the ganglion cells. They will eliminate the unwanted impulses by making a role in between the synapses.
But do they help in driving a car? Is there any role of amacrine cells while we
Yeah,
are
they will
driving?
detect the motion, no? So
Okay.
obviously they will help in driving a car.
Thank you, ma'am.
Teacher, we liked your presentation. Next time,
Thank you.
make it more attractive and we will enjoy that.
Okay, thank you.